Hello YouTube, welcome to our channel Biogan. In today's video, we will be discussing about zombies in biology. In our previous video, we had talked about the scientific aspects of zombies. The link of the video is also provided in the description box. By zombies in biology, we mean pseudoenzymes. The question is, what are pseudoenzymes? These are enzymes which have been predicted to have near zero or lack catalytic activity. These enzymes contain protein domains which resemble a catalytically active protein domain, for example, a kinase domain. These have been predicted to lack catalytic activity due to the lack of key amino acids or catalytic motifs. Pseudoenzymes have been formerly termed non-enzymes. How are pseudoenzymes related to zombies? These are considered to be zombie version of proteins. These are considered dead in the sense that they lack catalytic activity, undead as they perform important cellular functions. It is due to this relation with the zombies of being undead dead that these are known as zombie proteins or biozombies. Question arises, if we are saying pseudoenzymes to be catalytically inactive, then how do they function and play important roles in cellular processes? The answer to this lies in the fact that pseudoenzymes may have evolved to carry out different functions through other functional domains. For example, pseudokinases or pseudophosphatases employ at least one of the following mechanisms. They may act as modulators by serving as dimerization partners or cofactors, thereby inhibiting or accelerating other enzymes' activity through an allosteric transition. In cases where an enzyme exhibits a restricted subcellular localization pattern, the pseudoenzyme may serve as an anchor that traps a substrate to particular cellular locations and help in the functioning of other enzymes. They may competitively bind to substrates of other enzymes and act as competitors. The pseudoenzymes may function as an integrator where it integrates with the components of different signaling pathways or with multiple components of a linear cascade and thus lead to a biological function. Other classes of pseudoenzymes also follow one of these mechanisms. How are pseudoenzymes identified? This has been mainly done by sequence comparison. The first pseudoenzyme to be identified by sequence comparison was alpha-lactalbumin. This was reported in the Journal of Biological Chemistry. Alpha-lactalbumin bears sequence similarity to the enzyme lysozyme. Both these enzymes belong to the glycoside hydrolase family 22 of glycoside hydrolases. Glycoside hydrolases hydrolyze the glycosidic bond between two or more carbohydrates, or between a carbohydrate and a non-carbohydrate moiety. Lysozymes retain this function as it hydrolyzes the glycosidic beta-1,4 bond between N-acetylglucosamine and N-acetylmuronic acid. However, alpha-lactalbumin lacks this function and acts as a regulatory subunit of lactose synthase, helping in milk production. However, one should be careful while deducing a pseudoenzyme solely based on its sequence. This may lead to false discovery. False discovery can be avoided by verifying the sequence similarity with the structural and biochemical similarities. Where do pseudoenzymes come from? Pseudoenzymes may have evolved through gene duplication of their enzyme counterparts. The selective pressure at the catalytic motifs normally required for canonical enzyme function might have been absent, thus giving chance for the classical enzyme fold to repurpose or arrange in a different way, evolving new functions and leading to the formation of pseudoenzymes. However, one should not rule out the possibility that the enzymes might have evolved from their pseudoenzyme counterparts. The field of pseudoenzyme research is relatively very new, and new members are being added regularly. The field is very dynamic, and before concluding an enzyme or protein as pseudoenzyme, biochemical, structural, and functional aspects of it should be looked into very carefully. New and improved techniques and algorithms need to be incorporated. It might also be possible that pseudoenzymes might be new enzymes and their catalytic sites might have evolved to function on very different substrates, of which we do not know about due to the lack of in-depth experimentations. Thus, a long journey awaits before some recommendations about identifying a protein as pseudoenzyme can be generalized. Thank you for watching. For more such videos, please subscribe our channel Please like and share the video.